This is a University of Otago podcast.
All right, so we're going to have the next session, the next speaker in this session, I should say. So we've got Georgina Shillito and Sara Miller. Sorry, Shillito. Sorry, I got that wrong. And they're going to be going over the uses of Raman spectroscopy. Yeah.
Hi, thanks for the introduction. So today we're going to talk about something called Raman spectroscopy and how it can be used for a really diverse range of applications, such as detecting counterfeit medicines and then looking at new molecules for dye-sensitized solar cells. And so we both are based in the Department of Chemistry under Professor Keith Gordon. So what is spectroscopy? Spectroscopy is the study of light interacting with matter. And there are two sort of main interactions that we see every day, absorption and scattering. So we absorb these light-matter interactions when we look at the grass. We see that it is green, and it is green because there are molecules in the grass that absorb blue and red light and scatter back the green light, which we can observe with our eye. And it is this scattering the scattering phenomenon that we're going to be talking about today called Raman spectroscopy.
So as Sara said, Raman spectroscopy is a study of scattered light. So all molecules vibrate, but the arrangement of atoms and how these atoms are bonded together in the molecules determines the specific way in which the molecules can vibrate and what energies these vibrations occur. So in a Raman experiment, what we do is we shine a laser light on the molecule and then measure the scattered light off the molecule. So this scattered light has a slightly different energy because the molecule absorbs some of the energy from the original laser light. This causes the molecule to vibrate in a specific way with a certain energy. From there, what we can do is we generate a Raman spectrum. So this gives a measure of all the different vibrations that the molecule can have and gives the energies of each of these particular vibrations. So this here is the Raman spectrum of formaldehyde. So it's quite a simple molecule, so it's only got a specific number of vibrations. And this can give an idea of the molecular structure of the compound. Each molecule has a unique Raman spectrum, so this can be used like a molecular fingerprint, which is very useful in analytical chemistry in terms of identifying particular compounds.
So we use this molecular fingerprint to work out what the sample is made up of. And we can use this in applications like detecting counterfeit medicines.
And we can also look at it in terms of molecular electronics, where we can study specifically how the molecule interacts with light and try and use some of these properties to develop a dye-sensitized solar cell.
So the first application is counterfeit medicines. So there are three, besides detrimental health effects, legal issues and consumer trust issues, there are kind of three main problems that we get in counterfeit medicines. It can have too much of the active ingredient of interest. It could have too little of the active ingredient, which means that you don't get treated, or you could develop drug resistance, or it could have completely the wrong active ingredient present. In this example here, what happened was we had two of those three effects. We had the wrong active ingredient in too high a dose, so it was toxic. And these products were sold on just in the street, and the people who took these, their blood sugar bottomed out, which resulted in 11 deaths and 24 coma cases. Now, it's important to note that in New Zealand, counterfeit products aren't that big an issue. If you go to your local pharmacy, there should be no counterfeits. It's only when people are ordering products online from websites which kind of conceal where they actually are based, it can be counterfeit in over 50% of cases. Whereas in some countries overseas, such as India, if you were to go into the local pharmacy, it could be up to 25% of the products are counterfeited. So we got a series of products from MedSafe. These had been intercepted at New Zealand's International Mail Centre. These are products that people had ordered online. We have our initial or our genuine product. So this is the one that is the developer manufactures it. We also have some generics, so these have the same active ingredient and should have the same effects. And we also have counterfeits, and these ones are pretending to be the real thing, but they may not work in the way expected. So what we did was we took a series of our samples and we shine laser light at them, and we basically get a lovely fingerprint for each tablet. So this is an example of a fingerprint of a genuine product, and for example this peak here is associated with this part of the molecule, and so if we have different molecules or different ingredients in the tablet, this fingerprint will look different. We took a series of measurements from genuine products and found the small amount of variance between samples, which could be to do with how we focus the laser or slight variation in the batches. And we've got the small amount of error compared with the average fingerprint. We then can take any other or unmeasured products and compare that to them, and if they fit within these bounds, we can say, yes, that is the genuine product. But we can also then see if there's something that isn't genuine. So here, this is a counterfeit product, and you can see that the fingerprint is clearly nothing like that of the genuine product. But at least it does have the correct active ingredient in this one. And here's an example of another counterfeit product, and here you can see that it actually did not even have the correct active ingredient. Instead, it had this structure here, which is sildenafil, and it's the active drug in the competitor's product, Viagra. Now, we can also, because we had a large data set, we can kind of describe it in simplified terms by doing something called principal component analysis. Basically, this is where we look at all our samples and we compare them based on their variation. And so each spectrum or fingerprint is now represented as a single point in this space, with samples that have a similar fingerprint clustering really tightly together, and samples that are very different, separating quite a long way out. And if we look at this here, we see that, for example, genuine product with a 20 milligram dosage, we actually have 3 different batches and multiple tablets from each batch, all in this really tight area. And this is, highlights that the tablet to tablet and between batches, the quality control is really good in the genuine product. And in the green are some generic products, and we can see that they cluster quite close to the genuine products, and they also have quite tight clusters. So again, their quality control is reasonable. And we can also see the counterfeits, which are in red. They cluster quite a wee way away from your genuine products, and some of them have these really massive clusters. So this means that within the tablets and between the tablets in the same batch, they're extremely variable. And so this indicates that it really wasn't made in very good conditions. It probably was made in someone's garage and not mixed properly. And so using this molecular fingerprint, basically we could tell if a sample was genuine or non-genuine. We could tell if it had the correct active ingredient or not. And then we can also get an idea of if it's going to be subtherapeutic, therapeutic, or toxic levels of the active ingredient.
So in the molecular electronics unit of our research group, we look at instead of looking at the analytical systems, we look at the properties of individual molecules and how they specifically interact with light. So if these molecules have a useful or a particularly interesting interaction with light, we can try and optimize these properties for use in an application, such as a dye-sensitized solar cell, which aims to replicate photosynthesis by converting solar energy into electricity. So with a traditional solar cell, these are traditionally made with silicon, which is notoriously expensive, and the cells aren't particularly efficient. However, with a dye-sensitized solar cell, what we use instead is photoactive dyes combined with titanium dioxide nanoparticles that absorb the light from the sun. So because we can use a range of different molecules, these molecules have slightly different properties. So this means that we can make a range of dyes that have a range of different colours, and these are also far cheaper to make than standard silicon cells. So the main focus of the research in this area is to try and optimise the properties of these dyes to make a cell that is more efficient. And this is where Raman spectroscopy comes in. So as I showed you before, this here is the Raman spectrum of formaldehyde. So formaldehyde is a reasonably small and not particularly interesting molecule. So it has quite a simple Raman spectrum, whereas a compound that could be used in a solar dye, such as the one shown here, is a much larger, much more complicated structure, so therefore it has a much more complicated Raman spectrum. So this is where we need some computational chemistry to help us analyse the system and just understand what's going on. So what can we do with a computational model? Well, the first thing that we can do is we can model the molecular shape. So this here is the drug that Sara was talking about before. So when we model the shape, we can see how the atoms in this molecule are orientated relative to each other. From there, we can then generate a Raman spectrum and identify characteristic vibrations that can give us some idea about what is going on in the molecule itself. And lastly, what we can do is we can look at how the electrons in these molecules are moving. And this is perhaps one of the most important things in terms of designing something for a solar cell. So when you have a molecule in its original state, when it hasn't been exposed to light, it has a specific low energy. However, when it absorbs some sunlight, this causes the electrons in the molecule to move and gain energy. So the molecule is now in a higher energy state. So if you just leave this molecule, the electrons, after a specific amount of time, they relax back down and the molecule returns to its original state. So in a way, essentially, the light that is absorbed by the compound is kind of wasted. However, if we can find a way to couple this reaction to some sort of external circuit, we can utilise this energy to generate electricity. So where the molecule absorbs the energy and then the electron is transferred and can do work and generate electricity. So this is essentially the key component behind a dye-sensitised solar cell. So if we can use Raman spectroscopy to design or to understand the properties of compounds to try and optimise this process, in theory we can try and develop a solar cell with a greater efficiency.
So in summary, Raman spectroscopy gives us a molecular fingerprint of each molecule. And we can use this for analytical applications, such as detecting counterfeit pharmaceuticals.
And we can also use it with computational chemistry to try and understand how a molecule interacts with light and try and use this for a useful application.
And in our group, we've done many other interesting systems. So on the analytical side of things, we've also looked at things such as pigments and artworks. We've looked at foodstuffs such as cheese and milk, pharmaceuticals, geological or rock samples, and also biological samples such as teeth.
And in the molecular electronics side, we've also looked at some photocatalysts that would split water, for instance, to generate hydrogen and oxygen for fuel. We've also looked at some photovoltaics, LEDs, and other molecular sensors.
And finally, we'd like to thank Professor Keith Gordon and the KCG Research Group, along with James Orton and Claire Strong as collaborators as part of the Counterfeit Research Project. Thank you.
Thanks for that. Does anyone have any questions? So I have a question. So the herbal supplement market in New Zealand is unregulated.
Yeah.
Does Raman have the resolution to deal with such what would be a noisy product?
Okay, so actually there has been some work carried on from this to look at the herbal products, and maybe not necessarily, we've tried it with heaps of different spectroscopic methods, and the Raman is quite good at detecting the drug. So if there is a bit of drug adulterated in the herbal product, it can see it. But if you use other spectroscopic techniques, they also can pick it up. So it's kind of picking the spectroscopic method that will detect the drug and kind of not be so sensitive to the other stuff.
So if you know the active ingredient that you'd expect to see in a herbal supplement,
or I guess you'd want to use a couple of different techniques to make sure that you're definitely picking it up. Yeah.
Okay, cool. Thank you.